Everyone, and welcome. We're about to do a super high-yield immunology review designed to give you exactly what you need for the USMLE Step 1 exam and nothing you don't. We're going to cut right through the noise and focus on the must-know concepts that are absolutely going to score you points.
Okay, let's just dive right in with a question that trips up so many people. What is the real difference between a type 2 and a type 3 hypersensitivity reaction? I mean, they both involve antibodies, right? But in one, the antibodies are attacking a fixed target on a cell. In the other, you've got these little antibody-antigen complexes floating around, causing trouble wherever they land. Do you know which is which?
If that question made you pause for even a second, don't worry, you are in exactly the right place. By the time we're done here, you're going to be able to spit out the differences between these high-yield concepts like it's second nature. Let's get this stuff to finally click for you.
Okay, first up, Section 1, your immune arsenal. We have to master the fundamental players, the weapons and the soldiers, before we can even begin to understand the diseases. This stuff is the absolute bedrock of immunology.
And our tour has to start with the star of the show, the antibody. You can see it right here, this classic Y-shaped protein. It's built from 2 heavy chains and 2 light chains. But the real genius is its 2-part design. One part to find and grab a specific bad guy, and the other part to scream for help and call in the cavalry to get rid of it.
So let's break down that Y structure. The top 2 arms, that's the Fab, which stands for fragment antigen binding. Makes sense, right? This is the variable part, the part that gives each antibody its unique superpower to recognize one specific thing. Now the stem is the FC region, and here is your super high-yield mnemonic. Just remember the 3 C's. The FC region is constant, it binds complement, and it binds to cells. That's what determines its function.
And that FC region is what determines the antibody's class or its isotype. Here are the big ones you just have to know. IgG is your main workhorse, and it's the only one that crosses the placenta. The mnemonic is IgG greets the growing fetus. IgA is in all your secretions, tears, mucus, you name it. IgM is the megapentromer. It's the huge first responder on the scene of an infection. And finally, Ig is for allergy, and it helps fight parasites by activating eosinophils.
Next up is the complement system. Think of it as the antibody's special ops support crew. It really has 3 main jobs. First is opsonization, where C3b basically puts a giant eat me sign on pathogens. Second, inflammation, where C3a and C5a act like a flare recruiting other immune cells. And third, cytosis, where the membrane attack complex, or MAC, literally punches holes in bacteria. And listen up, here is a massive exam trap. If a patient is missing that MAC complex, they become super susceptible to recurrent Neisseria infections. Star that one.
Okay, so we know the tools in our arsenal. Now in Section 2, let's talk about how our bodies actually acquire this immunity. This is the whole active versus passive debate.
This distinction is so high yield. So passive immunity is basically getting a cheat sheet. You're given pre-made antibodies. It's super fast, but it doesn't last long. The classic example is a baby getting mom's IgG through the placenta. Active immunity, on the other hand, is when you learn the material yourself. Your body sees an antigen and figures out how to make its own antibodies. It's a bit slower to get going, but it lasts a lifetime because you develop memory. Think about getting a vaccine or actually getting sick and recovering.
And now for the main event. What happens when this incredibly powerful system goes wrong? We are now opening the case file on friendly fire. That's when the immune system gets confused and starts attacking things it really shouldn't.
So there are 4 main ways the immune system can overreact, and lucky for us, there's a fantastic mnemonic to remember them. Just think acid. Type I is A for anaphylactic. Type 2 is C for cytotoxic. Type 3 is I for immune complex, and type 4 is D for delayed. Let's break them down.
Okay, type I. This is your classic, immediate allergic reaction. It's all about IgE antibodies sitting on mast cells just waiting to get triggered. Think hay fever, asthma, a bee sting. Now type 2 is totally different. This is when our own antibodies, usually IgG or IgM, directly bind to antigens on the surface of our own cells, painting a target on their backs for destruction. Good pasture syndrome is a perfect, tragic example of this.
All right, so remember that question from the beginning? Here's the answer. In type 3, antibodies bind to soluble, floating antigens. They form these little clumps, these complexes, that drift around and get stuck in tissues like the kidneys, causing all sorts of collateral damage. Lupus is the classic example. But type 4? Type 4 throws antibodies out the window entirely.
This is a purely T-cell-mediated delayed reaction. Think about the rash you get from poison ivy or a positive TB test. No antibodies involved.
Speaking of autoimmunity, the USMLE is just obsessed with making you match autoantibodies to diseases. So let's do a rapid-fire round. Ready? Anti-dsDNA and anti-Smith? Bam, that's SLE. Anti-acetylcholine receptor? Myasthenia gravis. Antihistone? That's your buzzword for drug-induced lupus. Anti-TSH receptor? Graves' disease. And anti-glomerular basement membrane? That's our type 2 example again, Goodpasture syndrome.
So we've gone from friendly fire to full-on system failure. This is what happens when a key part of the immune system is broken, or was maybe never there to begin with. Okay, for your exam, this slide is maybe the single most important principle you need to know. The type of infection a patient gets tells you what part of their immune system is broken. If they keep getting lung and sinus infections from encapsulated bacteria, like strep pneumo or H. flu, your brain should immediately scream B-cell defect. But if the patient is getting weird fungal, viral, or other intracellular infections, you have to suspect a T-cell defect. It's all about pattern recognition.
Now let's hit 3 classic syndromes. DiGeorge syndrome is a T-cell defect from that 22q11 deletion. Just remember the catch-22 mnemonic. Wiskott-Aldrich is a combined BNT cell problem, and the mnemonic is water. Wiskott-Aldrich, thrombocytopenia, eczema, and recurrent infections. And finally, chronic granulomatous disease. This is a defect in your phagocytes. The key buzzword here is that these patients get sick from catalase-positive organisms, like Staph aureus. Why? Because those bugs can break down the tiny bit of peroxide the cell makes, leaving it totally defenseless.
Okay, for our final section, let's put these concepts to work in some very specific, very high-yield clinical scenarios: transplants and transfusions.
Transplant rejection really comes in 3 different flavors, and it is all about timing and the mechanism. Hyperacute rejection happens within minutes. It's a type 2 hypersensitivity caused by antibodies the patient already had. Acute rejection happens weeks to months later. This is mostly a T-cell-mediated type 4 reaction against the new organ. And then there's chronic rejection, which takes months to years, involves a messy mix of T-cells and antibodies, and ends in irreversible fibrosis. See how it all connects back?
All right, time to put it all together. Here's a USMLE-style case. A patient gets hives and their blood pressure drops. Anaphylaxis, just minutes into a blood transfusion. Now the key high-yield clue is the history of recurrent sinus infections. What's the link? Well, anaphylaxis during a transfusion is the classic presentation for someone with IgA deficiency. Their body makes IgE antibodies against the IgA in the donor blood. And those recurrent sinus infections, that's what happens when you're missing IgA, the main antibody in your secretions. So the answer is B.
Let's try another one. A little boy shows up with that classic triad: eczema, easy bruising because of low platelets, and constant infections. When you see those 3 things together, your brain should practically shout the mnemonic: W for Wiskott-Aldrich, A, T for thrombocytopenia, E for eczema, and R for recurrent infections. It is a textbook match for Wiskott-Aldrich syndrome. Answer C.
And for our last case, a young woman with a butterfly rash, achy joints, and kidney problems. I mean, that's a classic presentation for SLE. But the question asks for the most specific antibody. This is a classic USMLE trick. Almost every lupus patient will have a positive ANA, but it's not specific. Antihistone points you toward drug-induced lupus. The one that really locks in the diagnosis of SLE, the most specific one, is anti-Smith. That's your answer C.
And there you have it. We've covered the arsenal, the rules of engagement, and what happens when the system breaks down. By smashing these complex topics into high-yield facts and clinical cases, you now have the framework to tackle pretty much any immunology question they throw at you. You've totally got this. Now go crush your exam.